सुशील, ये उस वाले में डालना ना, मीट नाव वाले में ना, जो क्लास जो जो नया जो पासवर्ड जो बनाया है, मीट नाव करके। हाँ, एक लिंक नया डाला हुआ है। चलो मैं देखता हूँ उसमें कौन से वाले में आता है, देखता हूँ मैं अभी। एक मिनट रुको इसमें। अभी तो लिंक आया नहीं कोई सा भी। रुक रुक, लिंक इसमें तो आया नहीं, मतलब इसमें पासवर्ड वाला नहीं आया। एक मिनट रुक जाओ, दूसरे वाले में करता हूँ, दूसरे वाले में देखता हूँ। हाँ, पासवर्ड आ गया, ठीक है ठीक है। रुक जा। एल टी ए आई एम एस इंटर कर दूँ? हैं? हाँ, एम सी डालना है ना, कंटिन्यू डाल, इंटर कर दूँ ना? अच्छा, ये पहले उसमें दो जगह डालना पड़ेगा क्या पासवर्ड? हाँ, एक मिनट। एल टी ए आई एन। हाँ, हो गया हो गया हो गया हो गया हो गया। अच्छा, इसमें वो लोग तो नहीं करना अब? नहीं, जैसे वो मैंने तेरे को बताया था कि वो मीटर लॉक करना, जो वो तो करना है नहीं करना? च- ठीक है ठीक है ठीक है। हाँ हाँ, वो तो आता है, वो तो आता है। नहीं नहीं।
आपके तो पड़ी है, पेन ड्राइव पड़ी है। गलती से yeah, just is the audio okay now? I think I wasn't speaking actually. I was just setting up the system. Yeah.
Yes, your audio is correct.
Yeah. Yeah. So, good morning again. Last day of the year, and hopefully the next year brings better things for all of us with the COVID under control, and hopefully to see all of you in person when we are able to begin the routine classes in the regular way. So, today's lecture is on about cell membrane permeability and transports systems. So, allow me to begin with the topic. Yes. Go ahead. So the topic is the transport across cell membrane, but I'll give you a little quickly go through the physicochemical properties of cell membrane, because that is the barrier that has to be crossed by various solutes. A couple of things are repetition for you from perhaps your 12th standard knowledge. So let's begin with that. So why do we what is a membrane? Membrane is made up of a lipid bilayer with protein embedded in it. Good morning, Ashutosh. And it is not only a passive barrier but also have properties which makes it what we can call as an active barrier. And this is what we the picture to show you the latest model that we have for the cell membrane, what is called as a fluid mosaic model. Has this model been taught to you in 12th standard or earlier?
Yes.
Lovely. So a fluid mosaic model is the sort of latest model that we speak of, because Singer and Nicholson model. Before that there were other models. As people got more information, I'm just listing out some functions of the cell membrane. We just quickly have a look at these functions, just a compilation. So of course they form a selective boundary, limiting diffusion and creating a physiologically distinct compartment inside the cell. You do have membranes inside the cell around the organelles, but we will not call them as a plasma membrane or cell membrane. They will be organelle specific membranes, but the basic structure remains the same. And the membranes of course control the passage of electrolyte and small organic molecules. Because of their properties, they also have what we call as the resting membrane potentials, and which can then be utilized for the development of action potentials. They also provide surfaces for attachment of enzymes or various other signaling molecules, like reception of hormones and chemical signals. And they also serve as sites for recognition and attachment to the cell through various junctions that we spoke of in the earlier lecture. This is a minor compilation. It's not the exhaustive functions of the cell membrane. You can dissect each one of them out for further details. So what is cell membrane made up of? Answers:
Lipid and protein.
Lipid, proteins, and also a bit of layer of carbohydrate, what we call as the glycocalyx. So all the three major what we call nutrients, protein, carbohydrates and lipids, are part of the cell membrane. Lovely. So all three are present. Okay. So types of lipid present in the membrane. So you will find that majorly what we call them as phospholipids, sphingolipids. It's more of a little more of biochemistry, but majorly they are lipids. So they are large numbers, and the details perhaps somebody will cover details in biochemistry. I won't go into these, touching upon these components, but this listing of what we have. And this is a typical phospholipid which we find in the membrane. The structure on the left side shows that it's a glycerol with the two fatty acids attached to the C1, C2, and to the last one we have a phosphate group, and they were called phospholipid. It's lipid because you got two fatty acids attached to it, and depending upon the component which is attached to the phosphate group, we can call it phosphatidylcholine, ethanolamine, serine and inositol. And these four names you need to remember, because they are part and parcel of all cell membranes of all the cells, and all of them participate in some of the transduction function of the cell membrane, and I'll touch upon them briefly. Is this part okay with all of you? So the basic structure: the phosphate group along with the attached molecules like choline, ethanolamine, serine or inositol makes what is called as the polar head of the phospholipid molecule, and the two long chains of fatty acid form the non-polar tails. So I'm just showing you that how the arrangement physically would appear. So the molecular shapes could be in this particular manner, theoretically, structural organization of the 8 individual phospholipids. So the round is the polar component, and the straight ones are the fatty acid which is attached to the glycerol. So see how they can be arranged. So they can be a lamellar arrangement because everything is straight, nicely packed. Because the chains are moving away from each other, they will necessarily make it a curvature which is inside out, or an obtuse curvature which is a positive curvature, and the terminology is given: negative curvature or positive curvatures. So this is nothing but a physical arrangement which will happen on its own. Nobody has to think about it. And what we get from this diagram is that depending upon the nature of the phospholipid and especially the fatty acid chain attached to it, a membrane can take shape. So by changing the composition of the lipids in the membrane, theoretically the structure of the plasma membrane can be modified. But mind you that this is not the only requirement. It is a requirement, but the stability of structure is given by the cytoskeletal elements over and above these curvatures which can be introduced in the film. Will come to the proteins. So right now I'm just talking about the phospholipids as such. So the primary, what you can say, the component of plasma membrane is phospholipid, and phospholipids will arrange automatically in this manner. So a single bilayer will form a double layer, while a single layer will form a bilayer. The negative curvature will tend to form something like this, and the lower one inside out. So this is the arrangement, and this is arrangement purely because of physical interactions. It's just a name given. So just a name given, Gautam. So when it is arranged in this manner, where the curvature with the fatty acids chains outside, and the polar head curving inside, so that's called as the negative curvature. It's just a terminology. You could call it by any other name. You could call it inside out, outside in, which whatever terminology you want. So, but this is a consensus terminology. Or yes, you could. So you could have this is a physical arrangement. So if I were to take phospholipids of the top type and put it in water, they will auto arrange like this. They have no option. It's just the physical forces which will make them do this. Audio gone? Available? Not available? Let me mute and come back.
We can hear you, sir.
Okay. Okay. Yeah. Lovely. So if you gave me phospholipid in hand, okay, I had in a as a substance, and I put it in water, they will automatically do this. It's got nothing to do with thinking or biology. It's pure physical chemistry. They will do this, and they will not be able to be stable in this manner. Ultimately, they have to go to this kind of structuring. The non-polar heads will come together. The polar heads will go outside. So theoretically, these are the three types of combination possible. Okay, and let us see what makes them do that. So so here what we call as a micelle. Can you see the polar heads outside, and the inner core is form of the phospholipids. You have a lipid bilayer where the polar heads on the outside and the phospholipid, the fatty acid component, non-polar, on the inner side. And similarly, you can have a bilayer making an aqueous cavity. So again, what you realize is that because of the very nature of polar and non-polar components, and water being polar, the polar heads will be comfortable. I'm using an English phrase rather than the physical terminology. The polar components will be happy with the polar components. The non-polar components will be away from the polar component, and the only way that can be achieved in an aqueous solution is these couple of formations. So this is so if you took phospholipids, put them in water, this is what is going to happen automatically. Nothing required. So it's basically a self-assembling system. A self-assembling system. Its shape, its construction can be modified if you change the composition of the phospholipid molecules individually. So they need not be a very straight bilayer or a very nice spherical structure. It can be modified, but the stability may not be guaranteed. For stability, you would need cytoskeletal elements which hold them together. So what keeps phospholipid in such configuration? Because of the non-polar components wanting to be together, only one non-polar tail in micelle. See, in the micelle, the non-polar tails are coming together, so they are shielded away from the water area, the polar components. So all the polar heads go on the all sides in a sphere manner, so the central component becomes nice for the non-polar component. So yes, micelle structure, and this is what happens when you have fat and the fat is digested in the intestine. So this is what, and the bile basically comes and destroys this micellar arrangement, because you don't want micelle, they won't be absorbed. Okay. Now proteins are embedded in the lipid bilayer, and these are called as the integral proteins, and some proteins are attached peripherally or internally or from both sides onto this lipid bilayer. So you got two types of proteins which go through and through and which are on the sides. Now what's interesting is that if this particular protein which we are seeing on the slide, an integral protein, for it to be stably inside the fluid, because fluids phospholipids, so phospholipids arrange themselves but they are not held together, so they are not cemented, they are standing together, they are not cemented to each other, so they are mobile. The phospholipids can move around in the same bilayer, they can go around, and into this we have a protein now. Now this protein is also like floating in that phospholipid bilayer. For its stability in position, the parts of the protein, this the helix, the parts of the protein which have hydrophobic amino acids will stabilize in the central position, while the proteins with having the parts of the protein with the polar amino acids will tend to go on to the outer side. So all integral proteins will have a certain area which is hydrophobic in nature, and this is the area which will be stabilizing within the membrane. So the central part will be hydrophobic. These parts will have to be essentially polar, and this is how, if you give me a protein structure with amino acid sequence, by looking at the amino acid sequences we can conjecture that this particular protein is very likely to be an integral protein, even if we have no evidence. Only by the physical structure, the primary sequence, and if you have secondary and tertiary structure of proteins, one can make out whether this protein is likely to be an integral protein or not. Okay, so what is the role of the proteins? So this is one example to show how proteins can also influence the curvature. So this is a negative curvature on the other side. So there are two negative curvatures because of the arrangement, the spread out of the lovely Mahesh has not seen me. Okay, Mahesh, my name is. Okay, then who okay, there's somebody else as Mahesh, that's okay, as long as you listen to something and learn something good enough for us. Well, that's okay, Akram, as long as they're happy listening to us. Science is good for everybody, not just MBBS. Okay, so back to the ABC curvatures. So you see these there are proteins, the phospholipids marked as red, and you can see that their phospholipids are going away. Okay, they are arranged in that manner. So this will lead to a negative curvature on both the leaflets. So we call them a leaflet. So bilayer having a leaflet. Now this will not be stable. So A is not stable because both the curvatures are going away from each other. You can force it into bilayer, but it will automatically go away when the forces holding them together are gone. However, an hourglass shaped protein can relieve the curvature frustration. These terminologies I've picked up from textbooks. What is called as a curvature frustration. I mean, English coming into explaining as if the proteins are thinking. They are not. They are not frustrated. It's just a terminology to explain that the entropy is high. So the systems would like to have a low entropy, a stability, and when you force something against their physical orientations, it will develop potential energy and it will bounce back to its original A structure if not forced. So these proteins can help in removing these these the forced entropies and make the cells more stable. Now I'm giving you an example which perhaps you may have heard earlier. Okay, why leaflets tend to curve? Back to the slide. So I'll go back to this slide. So let us look at the blue one. You see the two tails which we are looking at. These are phospholipids. These are fatty acids, and fatty acids can be having a double bond or a single in between them, between the carbon. Whenever there is a double bond, it leads to a turning of the chain. So single bond will have a very straight arrangement. A double bond will lead to a curvature. So the top one has perhaps the fatty acids which are linearly arranged, all single bond, while the blue one has the two fatty acids having a double bond and leading to a kink in their orientation. As a result, they will go outside like this. Now when you want to pack such a thing, if I gave you a physical structure of the blue kind of arrangement, you will never be able to pack them straight, because they will tend to pack in this manner. So this is effectively because of the way the fatty acid chains, type of fatty acid chains, and their orientation is on the phospholipid molecule. So it is a physical structuring. The next question is why do the say I've got two phospholipids, why will they come together? Why can't they stay separate? That is because there's a polar component and there is a non-polar component, and polar component tends to come together with the water molecule, while the non-polar will go away from the water molecule, and this will in a way force the secondary structures like these, tertiary structure like these. So here the same arrangement has been shown. The arrangement number 2, blue, will not be tenable because the phospholipids are arranged with the tails surrounding with the water molecules. They will not be stable. Okay, so the only stable components are the top one in the brown and the one in the green. They are stable in aqueous solution because the polar heads are towards the water aqueous media, while the non-polar heads are together without having water around it. So non-polar and polar, this is what makes them have their structures and shape. Is that okay?
Yes.
Thank you. Right, Karthik. So let us look at the sodium channel. So a sodium channel is a integral protein which is in the membrane, and it allows the transport of sodium ions. Similarly, you got many other channels, potassium channels and calcium channels, and channels for anion and cations. So here you see, if you see the top left primary structure, so sorry, the secondary structure, you can see 4 domains, domain 1, 2, 3 and 4, and each domain has numbered S1, 2, 3 to 6. So each of those brown cylinder like structures represents one pass of the protein component. So this domain has got 6 transmembrane components. It's a single protein, mind you. It's a single mRNA giving a single protein embedded in the membrane. The central part of each of these transmembrane components will be hydrophobic, so that it is stable inside the membrane, and the peripheral components of each of the transmembrane components will be having polar amino acids, okay, with positive or negative charges. The central one will have, the amino acid will have no charges. And so this arrangement, so this protein has got 6 transmembrane domain, 4 domains with each having 6 transmembrane components, and the top shows as if it's linear. It's not. They will come together and form a pillar-like structure. So you have these 4 domains, and on the right is a 3D reconstruction of what we think this structure looks like. We have some data the scientists have acquired with various technologies, the actual arrangement, because this is not just imagination. This is based upon data as well. Domain is just a name given. So because this was very repetitive kind of the protein, so they called it domain 1, 2, 3 and 4, because the structure of domain 1 is very similar to structure of domain 2, 3 and 4 in its arrangement. However, the amino acid sequence of domain 1, 2, 3, 4 may be different. In fact, it is different. Okay. So that's just a nomenclature. You could call it somebody, but since the domain has been called, we are following it. So what you see is that these four colors represent each domain. They arrange themselves in this manner to make a hole in the membrane which is aqueous. This is the interesting part, because otherwise this membrane is non-aqueous. You have a polar component, you have the central hydrophobic component. There is no water hole in it. But these proteins, the way they come together, they make a hole in the center, which is aqueous. So therefore the proteins on the inner side, amino acids on the inner side will be polar. These will be non-polar. Am I okay with this component? That how integral proteins, columns of integral proteins, or the columns of transmembrane proteins of one integral protein. So this is one protein and with multiple columns, and they all come together and make a hole in the membrane. Is that okay? Lovely, Anaka. So this is how one can make aqueous pore through a phospholipid bilayer. There is no other way. You can't separate these phospholipids and assume that they will stable. They will not be stable. You have to put something in the phospholipid layer which can surround an aqueous pore. Okay. So this is the structure of all kinds of channels that we have that can pass polar molecules. Now look at the peripheral proteins. So this is a membrane shown below with the blue, dark blue chains. Single. I put only single one just to represent the no, they are not imaginary. These are alpha helix. So these are helices. Let me go back. So each domain forms one side of the pore. Okay, so this blue color, green and orange is one domain. So you got 6 cylinders forming one domain, and you got such 4 domains surrounding. Okay, and they form a channel. All right. So what you need is theoretically, even if you had 4 cylinders, not a pack of 6 as one domain, they can still form channel. So there are certain channels which do not have 6 transmembrane domains, 6 transmembrane components in one domain. There can be single transmembrane protein, and 4 of them can come together and make a hole in it, in the membrane. So each domain can be 1 TM, 2 TM, 4 TM or 6 TM. But what you need is 4 such independent components coming together. That's a requirement. So no matter what ion channel you look at, they will have the same kind of arrangement. The number of domains will have to be 4, but number of transmembrane in each domain will vary. So sodium channel always has 6, calcium channel always has 6, potassium channels, they are varied lot, so you can have 2 TM components, 1 TM, 4 TM, 2 plus 6 TMs, a huge number of variation is present for the potassium channels, but for calcium and sodium channels what we have is this kind of arrangement. Is that okay? I will come to the selectivity filter. I'll tell that. I'll come to that. But good that you noticed. So coming to peripheral proteins. So you have the lipid bilayer, and what you can see is through these something inserted in it, what we call the ionositol GPI anchor, and this linkage. So glucose, mannose, and something here I forgotten its name. Protein can be linked, and this is a way a protein is linked to embedded in the lipid bilayer. So the integral proteins were inserted in the membrane, while the peripheral proteins are attached to the lipid bilayer. And there are different ways of attachment. Terminologies are giving myristoylation, palmitoylation and prenylation. I will leave this to the biochemistry to touch upon this, but the concept I need you to remember: integral proteins versus peripheral proteins. Peripheral proteins directly do not engage with the lipid membrane, they will need something in between. So what you have here is a carbohydrate sort of an anchor, because mannose glucose, and they will link themselves to the membrane. They call GPI linked proteins, or directly to the lipids through these processes. Now the carbohydrates are attached to the membrane, and the way they are attached, they can be called as whether they are attached through O or through the N of the amino acid, they are called as O type or the N type. This is called glycocalyx, and glycocalyx is the layer on the outer side. So a cell membrane, so this is the, so what you are having is the lipid bilayer, proteins in it, proteins attached to it peripherally, and a carbohydrate layer over the membrane attached to the proteins, amino acids. So that's called glycocalyx. Now let's come to the component of membrane fluidity. Okay, Charul, I will tell you my name a little later. Let's finish the lecture first. Names are not important as long as you understand what I'm saying. Right. So membrane fluidity. So now, if you have packed lipids in a membrane, they are automatically arranged. Can they move around? Is the question. Can they move around? That's called membrane fluidity. So there are two factors which affect membrane fluidity: whether you have the double bond or the single bond, that is saturated fatty acid or unsaturated fatty acids. So the diagram on the right shows the arrangement of saturated fatty acid, the straight kind of arrangement, or unsaturated. Yeah, I know, I understand, Charul. I am getting that hang of the merits and demerits of the online teaching systems. I hope these two people are happy here to listen to what we are doing. So saturated fatty acids have a straight arrangement. On the right is the unsaturated fatty acid with a double bond, and there is a kinking in that. So if you have unsaturated packed, they are not very solidly packed, so they tend to have more fluidity, while the saturated are nicely packed, tend to have less fluidity. So the presence of cholesterol also helps out, and let me just show you another diagram to explain how cholesterol helps in packing. So I think I don't have a diagram. So what cholesterol does it? Cholesterol is a ring-like structure, so it can act like a packaging in between the phospholipids, and it being polar, it will be towards the central part of the plasma membrane. Maybe I have a diagram a little later to show you that. Next component, which is an important component, is membrane asymmetry. So you got two layers, two leaflets, the outer leaflet and the inner leaflet. So let me go back. So here, this is the membrane. You have one on the inner side and one on the outer side. So assuming that the lower part represents the cytoplasm and the outer part represents the ECF, you have this layer, what we call as the outer leaflet, and the inner leaflet of the plasma membrane. All of them have got phospholipids, but the type of phospholipid which is present in the outer leaflet and the type of phospholipid present in the inner leaflet are different, and this is what we call as the membrane asymmetry. So let's go to the membrane asymmetry. So asymmetry is primarily because of phospholipid distribution, and on the right side is a diagram. Percentage of total phospholipids. You see that both have, as total they are equal, but if you look at the sphingomyelins are more on the outer side, phosphatidylcholine is high on the outer side, while the phosphatidylethanolamine and phosphatidylserine is on the inner side. And very importantly, phosphatidylserine is not present on the outer side. This is a very sort of a important cue for the scavenging cells. If the phosphatidylserine goes on the outer side, it is an indicator to the scavenging cells like NK cells, the natural killer cells, to gobble up that particular cell. So a cell which is undergoing apoptosis, which is programmed to die, will, yes, correct. So it will expose the phospholipid onto the outer side, and then the rest of the system will take care of it. So it's interesting. So it's such a beautiful system that the way a cell will tell to the scavenging cells that I am ready for being eaten up is by just putting a flag of phosphatidylserine on the outer side. That's a beautiful signal. So you have asymmetry because of phospholipid distribution, asymmetry due to protein distribution because the peripheral proteins attached to the outer side and inner side can be different. You have glycolipids which can be asymmetric distribution, similarly cholesterol, sphingolipids and ceramides. There are different kinds of phospholipids. Ceramide is a different phospholipid. Sphingolipids are also phospholipids, and different cell types will have a different proportion of these. And later you'll get to know. The basic idea is to understand and appreciate the two leaflets are not mirror images of each other. They are highly controlled compositions of outer leaflet as well as the inner leaflet, and these processes are controlled by the genetic machinery, the enzymes which make these phospholipids. So now this is the latest model that we call as the Singer and Nicholson model, but there is certain data which does not fit these models, and I'll leave you to read this. But what we today call is a dynamically structured mosaic model. What it means is that the whole phospholipid layer is not uniform in the cell membrane. There are certain islands which are very stable. So what we call them the lipid rafts. So it is not that all the phospholipids can go everywhere, because cytoskeletal and the proteins, certain part of the phospholipid membrane are held together. So it is a mosaic which is dynamically structurable. So dynamically structured mosaic model, just a functional enhancement over the Singer and Nicholson model. So we are not changing the Singer and Nicholson model or the fluid mosaic model. We're just adding a layer of complexity to it, stating that the phospholipids are not just free to run around everywhere. They can run around, but in a limited way. Okay. So with this understanding of the lipid bilayer and various components, let's come quickly to the permeability and transport. So question: why should membrane be permeable? Because something has to be transported. What we need to transport in and out? Obviously ions, molecules which have to be glucose for energy, for fatty acid, vitamins, all sorts of things that you need. Whatever we eat basically must reach the cell and go inside the cell for it to be utilized. And whatever the cell has produced which needs to be thrown out has to go outside the cell. So there is no option but to have a cell membrane which is permeable. The problem, the two things that the cell have to encounter, the one is of course the lipid barrier. So lipid barrier was there to hold the components together, but it also must provide a way to allow these components to go in a controlled manner. Second is the issue of concentration gradient. So sometimes the ECF may have a very low glucose and the cell still needs it. So it has to pick it up from outside. So there has to be a mechanism, it can go against the concentration gradient, and we'll see how it happens. So got two kinds of processes: energy independent processes and energy dependent processes, and we'll take these two as. So energy independent processes. So all lipid soluble molecules and soluble can do this. So this is so a lipid bilayer is not a barrier to lipid. So lipid can go through lipid. So if there is a lipid molecule in the ECF domain, you could still ask a question that how is a lipid molecule stable in an aqueous medium in the ECF? That is also a handleable issue. We'll do it later. But assume that there are fatty acids roaming freely in the ECF and they come near a cell membrane. They'll just go through because lipids don't obstruct lipids. However, if it was a large aqueous molecule, it won't be able to go through. So all steroid hormones, large number of vitamins, nitrogen, carbon dioxide. Nitrogen is highly lipid soluble, it can go through. Carbon dioxide is also lipid soluble, and nitrogen carbon dioxide are very small molecules. So the main driving force of this diffusion, as if the membrane doesn't exist. So energy independent processes, one way is pure diffusion. The molecules are bumping around and they just diffuse out. So factors affecting diffusion, you all are aware of this: gradient, thickness of membrane, amount of area which can go across, temperature obviously, because more temperature, more Brownian motion, molecular weight of the molecule, heavier the molecule, slower the Brownian motion, and partition coefficient, which is nothing but the lipid solubility. More lipid soluble you are, easier it is to go through lipid membrane. Less lipid soluble, less you can go through lipid membrane. So the formula, you can just run through the formula. Okay. This is by the way, common question that teachers like to ask in the exam or the viva: factors affecting diffusion and these equations. So partition coefficient refers to the property wherein it basically reflects the proportional lipid versus the lipid permeability. So what happens when the molecules are lipid insoluble? What to do now? So herein we have Where is the square root? Square root, yes, square root. SQRT, square root of the molecular weight. Whoa. So when it is a lipid soluble molecule, no problem. No matter how big it is, it will go through a lipid barrier. No issues. So all the fatty acids, all the steroid hormones do not need a mechanism to go through lipid bilayer. They just pass through. Now the issue comes regarding lipid insoluble molecules. How will they pass through? So we need a mechanism by which they can be allowed through, and one of the mechanisms is called facilitated diffusion. And let us see what these mechanisms are. Facilitated diffusion. They are diffusing on their own Brownian motions along their own concentration gradient, but somebody helps them to get through. Okay. So one, we saw these channels which we had formed. This is an example of a potassium channel. So it is a channel embedded in the membrane as a combination of various domains we discussed earlier. The whole blocks form a channel in the membrane, and when this channel is open, potassium will just go through. So what we call diffusion through channels. In this case, okay, lovely. Okay, question: should channel be regulated? Will it serve some purpose, or they should always be open?
They should be regulated.
Lovely. So now, how to regulate? How to regulate? Should question is the why question of physiology. Yes, philosophically yes. Let's look at the physiology. How is it regulated? Voltage gated ion channels, ligand gated ion channels. So these channels, whether they are open or whether they are closed, can be managed by two ways: change in the voltage across the cell membrane, or presence of a ligand which bind to these channel proteins. So you can have acetylcholine gated ion channels. So we were discussing these I think in the nicotinic receptors, muscarinic receptors. They are nothing but ion gated channels. Do not all muscarinic channels are like channels. Some of them are not channels. Okay. So please note that many of the integral proteins to which ligand attaches are channels, but there are many other ligand attaching receptors on the plasma membrane which are not channels. They function in a different way. They function through enzymes for signal transduction. Okay. So example of voltage gated channel: sodium, potassium, calcium chloride. So sodium, calcium can be ligand gated also and voltage gated also. This is an example of voltage gated channel. So you also have a ligand gated channels. It's not that only voltage type occurs. This is an example of ligand gated acetylcholine gated cationic channels. So here you can see a different structure. So the voltage gated channels had 4 domain structure, while this particular channel has a 5 domain structure, but ultimately holding a aqueous channel in between, a hole in the membrane made up by these domains. Should the channel be selective? Again a philosophical question. Answer is yes. How to make them selective? And this is where somebody had asked me earlier regarding the pore selectivity, that if I have a hole in the membrane surrounded by domains, how can the channel be that selective that allows only sodium or calcium or chloride or potassium, or only cations or only anions? How is it achieved? Okay. So it's basically because of the amino acid sequence of the pore region. So in this diagram, if you see the 4 domains in 4 different colors, and you see this blue like a loop jutting in towards the channel area. Is it clear on the slides? These reddish round, it represents a single amino acid of the whole chain. Okay. So these cylinders are nothing but representation of it's a loop. It's a loop. It's basically a amino acid loop. So you got amino acid, and it forms a loop. And at the tip there is obviously there will be one amino acid at the tip of this loop. That is represented as these 4 maroonish or whatever color you are getting on your screens. Okay, what has been understood by research is that these 4 domains determine whether it will be a potassium channel or a calcium channel or a sodium channel. I mean, these 4 only. I mean, it's the beauty of the whole system. The rest of the protein structure determines the stability of the system. But whether it is a sodium channel or a potassium channel or calcium is determined by these 4 terminal amino acids on the loop. Okay, and experimentally, even if you change one of these 4, it can convert a sodium channel to a calcium channel. I mean, look at the selectivity. I mean, it's very precise. Okay, so evolutionarily, based on the amino acid sequences and the variety of potassium channel, what is assumed is that these, if you see on the left side, 6 transmembrane domain of one domain, transmembrane components of one domain, they come together to form, while on the right side is a potassium 1 domain. So this is 2 domain channel. Okay, 2 domain, while on the left it is 6 transmembrane domain. So I initially told you that it really doesn't matter whether you have 6 transmembrane domain. P is pore loop. P represents pore. So this is the pore loop. This which going out is called as the pore loop, because it goes towards formation of the pore controlling area. Pore is the hole in the membrane formed by the surrounded by the domains of the integral protein. All right. So to form a pore, you have to have this pore domain in the channel. So as long as I have this, TTX is tetrodotoxin. Tetrodotoxin blocks the channel, so it binds to the outer side, so TTX binding side. So if I want to block a sodium channel, I have to put a drug called TTX. It will bind to this and prevent the entry. And this is actually a poison, naturally occurring poison, which is few of the sea animals produce it and kill their prey by this. Because if you block sodium channels, all nerve transmissions are blocked. So the prey dies. So many sea animals produce this and they bite, they release this, and the animal which has been bitten gets these poisons and it blocks them and they die. And similarly, when we use local anesthetic, this is what they do. The local anesthetic will go and sit on these proteins, outer layers, and block the channel. So where they bind is called as the TTX binding site, or whichever. So you have a lignocaine binding site, or any other anesthetic, lignocaine or any other, will block sodium channel. And because sodium channel is required for action potential, there will be no action potential in the nerve. Basically, neural system is blocked. Okay. So this was diffusion through channels that we discussed. A channel only gave a hole. Diffusion occurred through that hole. There can also be another mechanism, diffusion via carriers. So now here's the slight difference. You have a integral protein which actually does not form a pore. There is no pore in it, but it can bind a solute on the outer side. When the solute binds, the protein undergoes a conformational change and then puts the solute on the inner side, or vice versa. Are we clear with the two? These channels form an aqueous pore through which the molecules go on their own diffusion gradients, on concentration gradients. While in the facilitated diffusion, there is no hole as such. There is the arrangement. The solute binds on one side, leads to conformational change, and it's exposed to the other side, and it goes out, whichever way. Okay. And here the selectivity achieved by the same lock and key model of enzyme and substrate reaction. So there is a structural arrangement to which it can bind. So this is the way selectivity of carrier is achieved. So both channels and carriers have selectivity. Of features of carrier mediated transport, one is of course specificity, which of course is also for the channels, what we call it the pore selectivity. Then saturation kinetics. What is saturation kinetics? That if I increase the amount of solute which has to be transported, the amount of transport goes on increasing but reaches a maximum rate beyond which it cannot increase. And this is what happens because of the insulin induced glucose receptors. So in the membrane, the glucose is transported through glucose receptors, which is a facilitated transport. So membrane has glucose receptors. Glucose comes from the ECF, attaches to the membrane, these proteins, glucose transporters, they undergo conformational change, glucose goes inside. Then it reverts back. So there is time required for this switching up. And therefore that limits amount of glucose which can be transported. So as you increase the glucose concentration, more gets transported, but then the limiting factor comes in, which is the switching rate of the carrier. In case of diffusion, because the channel is not participating in the process, more concentration gradient, more transport. So on the right side is the diffusion, rate of influx, ECF concentration, linear relationship. While on the left side is carrier mediated. You increase glucose concentration, the transport increases but reaches a Vmax, a maximum limit. So that's called as the saturation kinetics. The carriers and diffusion both are bidirectional depending upon the concentration gradient. Those the same blue transporters can also send it outside if the glucose inside the cell was higher. So this is a bidirectional thing. So we have energy independent processes we discussed, diffusion through lipid membrane, diffusion through channels, and diffusion via carriers. Please note I am using a very different terminology. Diffusion via carriers. They have to interact with the carrier. Diffusion through channels. Channels are just a gate. People move through on their own. Now let's come to the energy dependent processes where energy is required. What we call as the active mechanism of transport. Sometimes you need to transport something against the concentration gradient. So these mechanisms, you have heard these terms earlier, called calcium pump, sodium, potassium pump, and hydrogen pump. Okay, so ATP will be broken down and a particular ion will be sent across the cell membrane against the concentration gradient, simply use of energy, called primary active. And the most ubiquitous sodium potassium pump, it pumps sodium outside and pumps potassium inside. We know that the ECF has high sodium, so this is against the concentration gradient. Similarly, ICF has got high potassium, therefore this is against the concentration gradient, and you need ATP for this. It is an energy consuming process. I mean, all cells are alive because this particular pump exists. If this pump goes off, we all die. So features of primary active transport, ATP is used. It is also specific depending upon the pump that you have. So you have a potassium pump, you have a calcium pump, you have got hydrogen pump, and you got sodium potassium pump. It also has saturation kinetics. It has a limit to which it can work. It is unidirectional because it is direction specific, orientation depending. So if I wanted the sodium to actually come in, I have to put it in the reverse in the membrane. The same arrangement won't do it. Only one way gate. And it is electrogenic, this particular, because when calcium or sodium or anything is being pumped, you're actually moving ions across. So in the left side, two charges have gone in one calcium. On the right side, 3 sodium, 2 potassium, therefore 1 positive have gone outside. So these are electrogenic, right? Electrogenic. Okay, so these are other examples of what we call the ATP binding cassettes. They will do the same job. Let's go to secondary active. Now this is an example where the concentration, there is no actual ATP breakdown during this transport, but it's still active because energy of one guy's concentration gradient is utilized. So here we have a situation where sodium is high outside and sodium is low inside the cell. The glucose is low outside the cell and high inside the cell. How do we transport glucose into it? So we use the concentration gradient of the sodium. So the energy of the concentration gradient of sodium is utilized by this particular carrier to transport low glucose into the high domain area. Okay, this is called secondary active transport. What made the concentration gradient of sodium? The sodium potassium pump. So it's like a Paytm kind of stuff. You already charged your wallet and then you use the wallet, but your bank, so sodium potassium channel is your bank which generates that cash, and that cash is utilized through wallet to do something else. So the concentration gradient of the sodium is utilized for glucose transport but the sodium concentration gradient itself was produced by an active transport mechanism. All right. So there are multiple examples. When they go toward the same side, we call them symport. When they go opposite, we call them antiport. Mind you, not all antiports are secondary active. Some antiports are also very passive. Okay. So this is an example of a transport. Again, in an antiport manner, the concentration gradient of sodium is again utilized to pump potassium outside. The calcium outside. Okay. So the previous example was symport, the direction of glucose movement and sodium movement in the same side. This is the antiport, but antiport utilizing the concentration gradient of sodium. Okay. So three side by side, a uniport, a symport, and antiport. Okay. So with this, I think I will close my talk. Queries on this particular discussion? Let me put that slide back. Is ATP used? Okay. If it is, then active. If not, then inactive. Rate of diffusion through a channel. So imagine, if you remember, let me give an example of a rotating gate, okay, or a gate which is open. So if a gate is open, the number of people who can go through depend upon the push that you get from behind. But if it's a rotating gate, okay, then only one can go at a time. So the gate must come back to take you in. Therefore, it has a limitation as to how many people it can transport. In the same way, a carrier, because it needs to interact with the molecule to be transported, undergo a conformational change, and then revert back. It needs time, and therefore there is a limit to which it can do. On the other hand, a channel is opened up, you can go through depending on your concentration gradient. Channel doesn't bother. So that's why there is no limit to the simple diffusion, but there is a limit to the diffusion through a carrier, via carrier. Yes, symport may be active or passive. Yes, it can be.
So sir, diffusion through simple diffusion and channel will be same and carrier will be less than both of them.
That's correct. So you're correct that simple diffusion through lipid bilayer or simple diffusion through a channel, they have same kinetics. More the concentration gradient, more the travel. If lipid is not soluble in phosphate charge covering. Yeah. So right. So this is where you're correct that you're looking at a lipid and there is a top polar area and there is a lipid in between. So the major barrier is the lipid, not the polar. Okay. But yes, it is not. Had it been pure lipid, there would be no barrier. So the ECF lipids, Leela, I'll just come to you, but let me just handle what Kratik was saying. So you have an ECF lipid. Now let's look at a question that how is an ECF lipid in an aqueous medium? Is it floating freely or it has attached proteins? So almost all lipids in ECF will have some attachment to make them aqueous soluble. So for example, when you look at lipids, when you do liver function test, you talk about lipoproteins. What are lipoproteins? Nothing but proteins which attach to lipid. Then we need these proteins attached to lipids so that it makes it water soluble. Otherwise, you will have fat globules in your blood, because the lipids will come together and from a fat what do you call micelle. The proteins in a in a lipoprotein ensure that these lipids do not form that kind of a ball in the blood. Okay, so you need, so therefore so-called lipid and so-called free fatty acids which we speak of, which are floating in the ECF, do have certain attachments which make them aqueous soluble. Yes, like chylomicrons, all of them. Because see, it is theoretically, you take a phospholipid, put in water, it will form micelle, it will form globule. There is no option. But in your blood, when you eat food and the fat is absorbed, is it forming globules in your blood? Answer is no. Somebody is preventing them from forming those globules, and these are those proteins. So these proteins will help them get through that the outer polar zone, and then leave them. So steroid hormone comes with the protein, reaches the membrane, the protein stays outside, the lipid goes in. That's the way. But the if you Pratik, if you ask me the pure physics chemistry of it, I don't know. I don't have that kind of information. Maybe people have that, the exact physical nature of interaction and the various equations of physics or chemistry which make them pass through the outer polar zone. I'm not capable of answering that question, but your question is very valid. That if it, yeah. So thank you, Kritik. Back to somebody who's asking me, Mriganka, regarding the domains in sodium channel. Let me go back to the slide. Okay. Go back to the slide. Where is the slide? Yeah. So this is an example of a voltage gated sodium channel to sort of demonstrate how a pore is formed in the lipid bilayer. So what you have is on the right side an arrangement of green, blue, pink, and what is this color? Whatever color, forming the pore. Each of these colors represents one domain. In case of sodium channel, it is made up of 6 transmembrane protein components. See, these are 6 transmembrane zones. They come together to form one domain, and you need such 4 domains to come together to form a pore. For sodium channel, it is 6 transmembrane which form one domain. For potassium, it can be 2. You also have single transmembrane domain channels also, but they are coupled with 2 transmembrane component TMs domains. Is that okay, Mriganka? Yes, Gautam, yes. The secondary active transport, just 2 minutes. Secondary active transport will need a gradient to be created. Somebody has to create that gradient, and that is sodium potassium pump. So see, effectively all gradients are because of sodium potassium pump, and most of these secondary active transport are linked to sodium. I don't remember an example of something linked to potassium gradient. Almost all transport mechanisms of secondary active type are linked to sodium gradient. Right? Thank you very much. I had a wonderful time interacting with all of you. You are a bubbly bunch of lot, and stay like that for the rest of your life. Don't get old. Yes, Kritik. Yes, it is. So the Kritik, if you look at what is meant by conjugation, so conjugated bilirubin is conjugated to what? A glucose.
In the liver,
Yes, but conjugated to what?
glucuronic acid.
Correct. So that changes the solubility. That changes the solubility. But both are soluble.
Both are soluble. Okay.
Yeah. Their solubility changes. Amount. So it makes it more soluble.
So no, like if there is a direct question saying that unconjugated bilirubin is soluble or not, then
I would say trick question. I say it is soluble because you didn't ask me how much. So it's a qualitative question.
yes.
I mean, so because if I say a partition coefficient, I say it's partition coefficient 0.0001, but it is still soluble. Okay. So MCQs are not necessarily the best way to understand, because life is not plus or minuses only. Unfortunately.
Sure.
I mean, unfortunately, MCQs are the worst thing that has happened to humanity. And it's made us into options and this or that. There's nothing in between apparently in MCQs. So I'm not very fond of MCQs, though that's the necessity of today's education system and the large number of people that have to give exams. By the way, in the developed countries, MCQs are not the final way of selecting people. They're only for screening. So MCQs are only used for screening, not for final. Okay? Because they still believe that other components of life are more important than ability to remember or answer an MCQ. With this, thank you very much. All of you have a wonderful next year. I hope this year was good too, despite the pandemic. And the pandemic has taught us many more things about in life. That not just the education. Education will go on. वो चलता रहेगा, schooling चलती रहेगी. Just stay happy and follow your passions. Thank you. हाँ थोड़ा सा, हाँ ठीक है, इसको हटा दो, स्क्रीन हटा देता हूँ जी। अच्छा, दोनों म्यूट हैं, स्क्रीन और ये इन्हीं का ही है, मेरा नहीं। ओके। अरे ये भी तो पड़ा हुआ है। हाँ जी, लैपटॉप भी मेरा ही। चलो, थैंक यू।
सर ये हेयर बैंड आपका नहीं है?
वो मेरा ही है।
Hello everyone, good morning.
Good morning, ma'am. Good morning, ma'am.
How are you? Good.
Yes, ma'am.
तो जो यहाँ पे नहीं है, they are missing the cold, हाँ? जो दिल्ली का जो कोल्ड विंटर है, So you are missing that winter. Now it is at its peak. Okay. So now we are starting with the function of WBCs. Okay.
Good morning, ma'am.
Good morning.
I had one doubt.
हाँ बोलो.
मैम जो अनकॉन्जुगेटेड बिलिरुबिन होता है, is that insoluble or soluble?
It is not. It is lipid soluble. It is not soluble in water. So to make it soluble in water, it is conjugated with glucuronic acid in the liver. Once it
Thank you
gets
so much.
conjugated, then it will become soluble and then it can be secreted in bile. Okay.
Okay. Yes. Thank you.
Okay.
But ma'am, albumin bounded unconjugated bilirubin.
Unconjugated.
Albumin bounded.
हाँ हाँ. Albumin binds with unconjugated bilirubin.
That's soluble in water.
Okay. So now let's start with the functions and fate of WBCs. Okay. So you all know that the function is to provide immunity to body. Okay, to provide immunity against bacteria, viruses, parasites. So how these WBCs perform their function to provide immunity? Now let us see. So have you received the mail regarding what are the rules to be followed during online lecture?
Yes, ma'am.
Okay. So if there is anyone which is joining which is not from AIIMS, not AIIMS student, so you have to notify it. Okay. So now let us start. Now as the out of WBCs, granulocytes are more, neutrophils are more common, so we are, then their number is more, so we are starting with the neutrophils. Now let us see about the lifespan of WBCs that we have already discussed. Okay, so granulocytes, they stay in the circulation for 4 to 8 hours and then they move to the tissue, and in the tissue they can live for 4 to 5 days, and after that there will be phagocytosis of these granulocytes. Okay, now for monocytes, they will stay in the circulation for 10 to 20 hours, and once they enter into the macrophage, once they enter into the tissue, they get converted into macrophage. Okay, so macrophage means their size will increase. Okay, changes will occur in their structure, and then it is labeled as the tissue macrophage, and then tissue macrophage can remain in the tissue for months. Okay, and the macrophages, these are the most strong, means they are more they are involved in phagocytosis of bacteria and also large particles. Okay, then lymphocytes. So lymphocytes, they are synthesized in the lymphoid tissue, in the lymph nodes, in the spleen, then they enter the circulation via lymph. Okay, so while lymph they enter into the circulation, again they move to the tissue. Okay, again they enter the lymph nodes, and again they via lymph they enter the circulation. Likewise, there is recirculation of these lymphocytes in the circulation and in the lymphatic fluid. Okay, like they provide the immunity to the tissue, and their lifespan is from weeks to months. Now the platelets are replaced every 10 days, so their lifespan is around 7 to 10 days. So to maintain the constant level of platelets, number of platelets, the body has to synthesize around 30,000 platelets per day. Now what is the function of some neutrophils? Are the most common cells in the peripheral blood. So how these neutrophils perform their function? Now out of all these cells, neutrophils and macrophages, these are the main phagocytic cells. Okay, eosinophils are also phagocytes, but they are weak, so that commonly they release the granular content and they kill the parasites. Okay, but the neutrophils and macrophages are the main phagocytic cells of the body. Now you know what is chemotaxis. Okay, so chemotaxis is movement of these inflammatory cells, immune cells, from circulation to the tissue. Now how this takes place? So for chemotaxis we need some chemical that is called as chemotactic agents. Now this as written here, so these chemotactic agents can be the bacterial or viral toxins or the degenerative products from the tissue or complement product of complement system. So we will discuss tomorrow what is complement system. So right now please remember that there is one system called as complement system, which helps in chemotaxis of immune cells from circulation to the tissue. Okay. Also there are plasma clotting products which will help in chemotaxis. Now here you can see in the figure that this is the center from where chemotactic material is secreted or released from into the tissue. So these neutrophils are present in the circulation, and then they travel across the endothelial layer and they enter the tissue space. Now that chemotaxis takes place along the concentration gradient of this chemotactic substance. Okay, as the concentration is maximum at the source, they move along the concentration gradient and they reach the tissue where they are required. Okay, in case of injury, okay, there is damaged material is there. In case of infection, the virus or bacteria or parasite is present there, so they will reach where they are required. Okay, so this chemotaxis is effective for around 100 micrometer distance. Okay, and most of the tissue is present, the distance between the capillary and tissue is only around 50 micron. So it is very effective mechanism. It is important because these WBCs are present in the circulation. Okay, in the tissue few leukocytes are present. So when there is increased need of these leukocytes, it is important to meet this need. Okay, so they more and more WBCs they can accumulate at the place where they are required to provide the immunity. Okay, so this is chemotaxis. Now what are the mechanisms for chemotaxis? So there are steps. The first one is rolling, then stable adhesion, then transmigration along this endothelial membrane. Okay, now let us see one by one. Now this is the neutrophil on left side. Okay, now there are certain adhesion molecules present on the surface of this neutrophil. Okay, now what are these adhesion molecules? So these are the selectins and integrins. Now we have three types of selectin. L type selectin is present on the surface of this neutrophil, and P and E selectin, they are present on the surface of the endothelial cells. Okay, now normally also there is some association and dissociation between these selectin molecules when there is no any inflammation or there is no any infection. Okay, but that association and dissociation constant is so high that there is no any permanent adhesion between endothelium and neutrophil. Okay, so they roll around along the endothelial membrane. Okay, so that is called as rolling. Now if there is any injury or if there is any invasion by the microorganism of the tissue, now this is a macrophage which is present in the tissue, that is tissue macrophage. Now the tissue macrophage will engulf this microorganism. Okay, and it will release the cytokines. Now we will see in detail how does macrophage function. So macrophage will release the cytokines and chemokines. So cytokines which are released from macrophage is interleukin 1 and TNF, that is tumor necrosis factor. Okay, now these cytokines, they will act on the endothelial cells and they will increase the expression of P selectin and E selectin. Okay, also the macrophage will release the chemokines, and these chemokines, they act on the neutrophil and they will increase the expression of integrins. Okay, so now here the expression means they are initially they are in low affinity state. That means the affinity of integrin with adhesion molecules on the endothelial surface is less, but once the chemokines act on these neutrophils, the affinity will become high. Okay, so here you can see the open ends, here the ends are closed. Okay, so the affinity between the endothelial cell and neutrophil will increase, and with the help of other adhesion molecules like intercellular adhesion molecules, which is called as AIIMS. Okay, so they will bind with the endothelial cells very tightly. So that is called as stable adhesion between endothelial cell and neutrophil. Okay, and then it is followed by the transmigration. So initially small portion of the neutrophil will protrude through the capillary membrane. Okay, and like this, because of this amoeboid movement, the whole cell will move from the circulation. So this is the circulation. Okay, this is blood vessel, and this is the endothelial lining of the blood vessel, and this is the tissue. So from this blood vessel, the neutrophils will enter inside the tissue. Okay, and in the tissue they will move via the amoeboid movements. They have actin myosin molecules inside them. Now what is actin myosin molecule you will learn in the skeletal and smooth muscle. Okay, so these are the cytoskeletons. They will help the cell to move. Now they will cause a movement of the cell, and then it will enter inside the tissue and towards the area from where the chemokines are released into the circulation. Okay, once they enter in the tissues, now they will perform their function, that is phagocytosis of the microorganism. So they will help macrophages, or they will work together with the macrophage to kill the microorganism which has entered the tissue. Any doubt? This is very important. Now how the neutrophils or eosinophil will also follow the same pathway. Okay, all the cells will follow the same pathway, and they will enter from the circulation to the tissue. Okay, because whenever there is any infection, suppose there is injury to the to your finger. Okay, so the organisms will be present in the tissue. Okay, so the WBCs have to move from the circulation to the tissue to perform their function. Okay, now so this is a schematic diagram of neutrophil. Okay, now it is showing molecular changes which are taking place in the neutrophil at different phases. So during the rolling phase, okay, so the neutrophils are not activated, so the calcium level is low. So calcium is stored in the intracellular storage form. Okay, now integrins on the cell membrane, they are low in number, while the selectin will be higher. Okay, because this selectin, L-selectin is present always. Okay, and because of this L-selectin, there is association and dissociation between the endothelium and the neutrophils. So they are present in close proximity with the endothelial cells. So it is called as rolling. Okay, now the oxidized components, they are dispersed in the cytosol. So yellow are the oxidized components. So they are present in the cytoplasm. Now we will see what is oxidized components. So it is NADPH oxidase enzymes and other components. Once the during transitional phenotype, okay, that is when there is activation, when the chemokines will act on the neutrophil, then what changes will take place. So during transition phase, again calcium level will be low, but the integrin level will increase in the cell membrane, selectin level will decrease, and oxidized components, they will move to the plasma membrane. Okay, now in the motile phagocytic type, the calcium will be released from the intracellular storage. Okay, and also calcium will enter from the outside, from the interstitial fluid. Also integrin level will be higher, and other adhesive molecules will be expressed on the cell membrane, so that there is strong bond between the endothelial cell and neutrophil will form. So these oxidase enzymes, they will activate it, and the metabolic and secretory responses are activated to release the granules outside as well as inside the cell. Okay, now what will happen? Now let us see. Okay, so these are the chemoattractants. Complement system is involved. So the C5a component of the complement system is chemotactic agent. Now leukotriene, B4 peptides, bacterial peptides, platelet activating factor, and interleukin 8. So all these are the chemoattractants which cause movement of these neutrophils and eosinophils, monocytes to the tissue. Now what is opsonization? Okay, this term we will use this term repeatedly. Now you know that there is phagocytosis of the pathogen. Okay, now how the neutrophil or macrophage will know that which whether they have to phagocytose that particle or not. Okay, whether it is self, tissue or the foreign substance. Okay, how they know? So for this we have different mechanism. Okay, now there are opsonins. So opsonization means it is a process by which a pathogen is selected for phagocytosis and destruction. So this process is called as opsonization. Okay, now there are different opsonins. Opsonins means these are the molecules which will bind with the microorganism, okay, which is to be phagocytosed. Now there are antibodies. Okay, antibodies, these are nothing but these are the proteins, plasma proteins, okay, which will bind with the antigen. Now what is antigen, antibody, we will discuss tomorrow. So there are IgG antibodies, complement components C3 and oligosaccharides terminating in mannose. So these are the substance molecules which will bind with the pathogen. Okay, they will bind with the bacteria or virus, and they will make sure that that virus or bacteria will get phagocytosed by the neutrophil or macrophage. Okay, so they will increase the probability or chances of these microorganisms to get phagocytosed by the phagocytic cell. That is called as opsonization. Okay, that means that pathogen is selected for phagocytosis and destruction because of these molecules. Now what is phagocytosis? So it is the destruction of the it is the intake and destruction. So there are different stages of phagocytosis we will discuss. Now how they will determine, how the neutrophil and macrophage will determine whether to phagocytose it or not, because we have so many tissues and other cells in our body. Okay, now it depends upon the surface of that tissue. Now the intact surface, the living surface is the living tissue will have the smooth surface. Okay, and if there is any injury or if there is any pathological invasion of the microorganism by that cell, then the surface of that cell will become rough. So if the surface is smooth, there will not be any phagocytosis, and if it becomes rough, then it will lead to phagocytosis. Now also our cells have some coating of proteoglycans, so that is a protective coating, and that will repel the phagocytes from their surface. Okay, and also opsonization. Suppose the antibody is binding to the any particle, suppose there is any tumor cell. Okay, so the tumor cell will also express some antigen. Okay, now or the surface of the tumor cell will be rough. So in that case, that tumor cell will also be phagocytosed by the macrophages. Okay, so what are the phases of phagocytosis? This is very important. Okay, so you have to remember all the phases of phagocytosis. Now this is neutrophil. Okay, now this is a microorganism or any other particle which binds with the surface on the neutrophil. Okay, now once it binds with the surface, the cytoplasm, these are the pseudopodia which is formed from the surface of the neutrophil. Okay, they will n number of pseudopodia will be formed, and they will fuse on opposite surface. Okay, so these pseudopodia will fuse on the opposite surface. Okay, and then they enclose one cavity in which there is presence of microorganism or any foreign particle. Okay, so that's why it is called as phagosome. Okay, so this is the phagosome or phagocytic vesicle which is enclosed into the lipid bilayer membrane, and it has the microorganism or any foreign particle or the material which will be phagocytosed. Now once it forms inside the cell, it will fuse with the lysosomes. Now in the lysosome we have various digestive enzymes. So we have proteolytic enzymes, lipases. Okay, neutrophil will not have lipase, neutrophils will have proteases. So these are the proteolytic enzymes. So they will cause digestion of this microorganism. Okay, now all the microorganisms will not be digested by the proteolytic enzymes. Okay, because some in the next year in the microbiology biology you will learn that some organisms have the protective coating around them. Okay, and that coating will not be digested by proteolytic enzymes. Then we have another system which is called as oxidase system. Okay, we will see. So that oxidase system is very powerful and it will cause it has bactericidal effect. They will kill the bacteria. Okay, now once it is digested in this phagolysosome, the residual body that will be removed from the cytoplasm. Okay, that will be excluded from the cytoplasm into the interstitial fluid. Okay, so these are the various phases of phagocytosis. Now the lipases are not present in the lysosomes of the neutrophils. Lipases are present in the macrophage. Okay, now because they are important because the tuberculous bacilli, they have the lipid coating around them, and to kill that tuberculous bacillus, it is very important to have the lipase. So macrophage will play very important role in the phagocytosis of this tuberculosis bacilli. Okay, and it is means every time it is not possible that the microorganisms will get killed by these phagocytes or by these neutrophils and macrophage. So some organism will survive this process and they will lead to chronic diseases like tuberculosis. Okay, why we develop tuberculosis? Because our microorganisms, they are not able to kill that bacillus, because they will secrete some enzyme to prevent this phagocytosis, or they have the protective coating around them. Okay, so if this happens, then they will remain inside the body, they will proliferate inside the cells and they will form the tubercles. Okay, that is a chronic disease. You will learn it in pathology, what is the pathophysiology of tuberculosis. Okay, so but the common bacteria and viruses, they will be destroyed by the these phagocytes. Okay, and they will remove these organisms from our body. Now what is respiratory burst? So we have seen that in lysosome, the lysosomal enzymes are liberated, also peroxisome enzymes are liberated, and they cause digestion of this microorganism. But when it is not sufficient, we have another system, and that is called as oxidase system. So during the phagocytosis, there will be increased uptake of oxygen by these neutrophils. Okay, so it is called as respiratory burst. Okay, because there is increased uptake of oxygen by these neutrophils. Now what happens during this process? So this oxygen will combine with NADPH with the help of NADPH oxidase enzyme. Okay, and it will form the hydroxyl ions. Also we have the superoxide dismutase, which will convert these hydroxyl ions into hydrogen peroxide. Okay, so these hydroxyl ions, hydrogen peroxide, superoxide. Okay, so these are all oxidizing agents. They will oxidize the contents of the bacteria and they will kill the bacteria. Okay, now we have another enzyme that is called as myeloperoxidase. Now what is the function of myeloperoxidase? It will catalyze the reaction between chloride ions or bromide ions with the hydrogen peroxide. So it will form the hypochloric acid, hypochlorite. Okay, and it is very powerful antibacterial agent. So it will kill the bacteria. So all these mechanisms can be divided into two types: oxygen dependent and oxygen independent. Okay, So the whatever oxygen dependent are again of two type, myeloperoxide mediated, that is formation of hypochlorous acid, and myeloperoxide independent. Okay, so up to this step that is myeloperoxide independent, and after this step we have myeloperoxide dependent. Why it is important? Because in some genetic diseases we have the deficiency of myeloperoxidase. Okay, and if there is any deficiency of myeloperoxidase, the neutrophils can cause phagocytosis, but because we lack the hypochlorous or hypochloric acid, the phagocytosis is the phagocytosis is not that much powerful. Okay, but they are able to do phagocytosis. Okay, so myeloperoxide is independent antimicrobial system is formation of hydroxyl ions, formation of hydrogen peroxide, formation of superoxide ions. So all these are oxidizing agents. Okay, so they will oxidize the contents of the microorganism like lipids, proteins, and they will cause destruction of this microorganism. So all these mechanisms are oxygen dependent. Okay, but in some tissues, like if there is formation of pus, okay, what is present in the pus? Why there is white color to the pus? Because of the high number of the neutrophils inside that cavity. Okay, so in pus we have so many neutrophils, but oxygen will be deficient because blood supply is not there in the inside that cavity. Okay, now in that case we also have the oxygen independent mechanisms. Okay, so whatever the contents are present in the lysozyme, okay, or these are the acids, lysozymes, lactoferrins, defensin, and permeability increasing proteins. So they will act on their own, so they don't need oxygen for the killing of the bacteria. Okay, so in the cavities where the oxygen tension is less, they will act and they will cause phagocytosis. Is it clear? Okay, so in which conditions the phagocytosis is defective? So in agranulocytosis the number of granulocytes is less. Then in congenital myeloperoxidase deficiency, in that case also we have defects in phagocytosis. Then deficiency of G6PD enzyme, neutrophil hypomotility. So in all these cases the phagocytosis will be defective, and in these patients we observe the chronic granulomatous diseases because the phagocytosis is defective. So the bacteria will survive inside the body and they will multiply inside these cells and they will form the formation of granules. Okay, so they are called as chronic granulomatous diseases, and this the most important example of this chronic granulomatous disease is tuberculosis, which is very prevalent in our country. Okay, everyone gets infected with tuberculous bacillus, at least in India. Okay, because the prevalence is so much and it is transmitted via respiratory droplets. So everyone will get infected by this tuberculosis bacillus, but only few will develop the disease. Okay, because once the bacillus enters inside our body, our neutrophils and macrophages, they will cause phagocytosis of this bacilli and the disease will not develop. Okay, but in few patients in which the immunity is depressed, okay, or if the phagocytosis is not effective, then they will develop the disease. Now what is neutrophilia? Neutrophilia is a condition where the neutrophil count increases more than normal. Okay, normally 50 to 70%. If it is more than 70%, then it is called as neutrophilia. Now what are there are certain physiological causes, okay, like in exercise, increased activity, injection of epinephrine, pain, nausea, vomiting, then pregnancy, menstruation, labor, injection of steroids. So all these are conditions in which the physiologically there is increase in neutrophil count. So you are not, so whenever you see any report, blood report, so you have to take into account all these factors also. Okay, whether the patient has performed any exercise before collecting the blood, or if it is a female, then whether she is having any whether she is pregnant or having menstrual cycle or any. Okay, so you have to consider all these reasons why there is increase in neutrophil count in healthy person. Okay, now what are the clinical?
Excuse me,
हाँ बोलो.
ma'am, please explain function of G6PD in phagocytosis.
So G6PD is reducing agent. Okay, so means we have seen that is it is required for the formation of glutathione. Okay, and the in the biochemistry you will learn in detail what is the these oxidizing agents and reducing agents and what is their function. Okay, and what is the what is antioxidant, that you will learn in detail in your biochemistry classes. Okay. Now what are the pathological causes for neutrophilia? So acute pyogenic infection, you all know that whenever we have any infection, so neutrophil count will increase, and also more specifically in bacterial infection. Okay, now in inflammation, if you have any tissue injury or trauma, in that condition that locally there will be inflammation. Okay, and in that case also neutrophil count will be higher. Now which are the conditions in which there is neutropenia? That is very important, because if you have typhoid or paratyphoid fever, so what we expect in fever, we expect most commonly it is a bacterial or viral infection. Okay, in which case there is increased neutrophil count. But if the patient has fever and neutrophil count is low, then you have to consider these possibilities, that there can be typhoid or paratyphoid fever or viral infection or bone marrow depression. Means in bone marrow depression there will not be fever, but this typhoid, paratyphoid and in viral infection most commonly we have increased lymphocyte count and monocyte count. Okay, so these are the condition in which neutropenia is there, and also if there is bone marrow depression because of any reason, because of the radiotherapy, chemotherapy, so in that case the formation of neutrophil will be affected, and so the count will be low. Okay, so that is all about the function of neutrophil. So the neutrophil will cause its function, so that it will go to the tissue and it will cause phagocytosis of microorganism, and during that process only it will get damaged and it will be phagocytosed by the macrophage. Okay, so that is about the neutrophil. Now what about eosinophil? So eosinophils, these are the weak phagocytic cells. So their main function is not phagocytosis. They release granules, okay, containing there are various factors which are present in their granules that they act on the, they are mostly important against parasitic infestation. Okay, so 3% of marrow cells are eosinophils, and in blood they will stay for 1 to 8 hours and then enter into the tissue, and in the tissue they can stay for few weeks. Okay, and their synthesis is regulated by the interleukin 3, 5 and granulocyte monocyte colony stimulating factor. Okay, so these eosinophils are present most commonly in the GI tract, okay, respiratory and urinary tract. Okay, so where are the so there are chances of entering the parasites from these places. Okay, so they are present along the lining of these tracts. So again the eosinophils also follow the same steps for transmigration into the tissue. Okay, and once they enter inside the tissue, they either accumulate as quiescent cells, or if there is any requirement, then they will cause release of their mediators from the granules. So eosinophils have 5 types of granules. Okay, but the most important is crystalloid granule. Okay, because how we observe, how we identify eosinophils from the peripheral blood smears? What are the characteristics of eosinophils?
Brick-red cytoplasmic color.
No.
Bilobe
Bilobe nucleus, and what is observed in the cytoplasm?
granules.
So granules, and these granules are brick red granules. So these are the brick red granules, and the granules are so many in number. Okay, so the cytoplasm is filled with the granules. Now out of all these granules, the most important granules are crystalloid granules. Okay, now why they are called as crystalloid granules? Because they have the crystalline core which is formed by the major basic protein. Okay, now this major basic protein is very toxic. So whenever it is present in the granule, it is in the inactive form, and it is present at the center in the as a crystalline core. Okay, now in the matrix of this core, matrix of this granule, we have eosinophil peroxidase, eosinophil cationic proteins, eosinophil derived neurotoxin. Okay, so all these are bactericidal proteins. They will attack the helminths. Okay, so parasitic larvae, they attack these parasitic larvae, and they will attach these eosinophils will attach firmly with these parasitic larvae, and they release these contents into them. Okay, now they are major basic protein, and all these contents, they are very toxic to these parasites, and they cause the formation of holes in the plasma membrane, and they will increase the permeability of this plasma membrane, and they will cause the death of this parasite. Okay, so as they are very toxic, they can cause destruction of the normal tissue also. Okay, so that is why we observe the damage to the alveoli and airways. Okay, in asthma patients, we observe large number of eosinophils, because they are involved in allergic reactions also. Okay, eosinophils are involved in allergic reaction also. So asthma is an allergic condition, and we observe large number of eosinophils which are lining the airway epithelium, and this major basic protein can cause damage to the healthy tissue also. Okay, so these are all the mediators which are released by the eosinophils. So large number of interleukins, cytokines, and growth factors also. These major basic MBP is the most important one. Okay, and all these contents will be released into the tissue once the eosinophils get activated. Okay, so these are the mediators. You need not there is no need to remember all the names, but you should know what is major basic protein. Okay, and it is released by the eosinophils and others few names. Okay, now in which condition we observe increased eosinophil count? So these are the infections, mostly parasitic infections. Also in allergic conditions like asthma, atopic dermatitis, gastroenteritis. So in all these conditions, in atopic dermatitis, the eosinophil will invade the skin and mucous skin. In gastroenteritis, the GIT. Okay, so they are involved in all these allergic conditions. Now whenever there is any drug reactions because of aspirin, sulfonamides, cephalosporin, there is a role of eosinophils in these drug reactions. Okay, now in which condition there is eosinopenia, that is eosinophil count will decrease. So the most common important is because of the steroids. Okay, whenever because of this, in bronchial asthma also we give inhalational steroids, and they are very useful, especially in patients in which there is increased eosinophil count. So if we have the patient of asthma, then we need to check the eosinophil count in the airways as well as in the blood. So if there is increased eosinophil count, then they are most likely to respond to the inhalational steroids. Okay, because the steroids will decrease the number of eosinophils. And also again in bone marrow depression there will be decrease in eosinophil count. Okay, so that is about eosinophil. Any doubt? Okay, so next one is monocyte and macrophage. So you know that is monocyte and macrophage. Monocyte is the cell which is released into the bone, into the circulation, and once the monocyte moves to the tissue, there will occur changes in this monocyte and it will be converted into macrophage. So macrophage is the more phagocytic, it is a strong phagocytic, powerful phagocytic cell in whole body. Okay, so the main functions of these macrophages are digestion of microorganisms and particulate matter. Like if there is any damage to the tissue or injury, then that matter will also be phagocytosed by the macrophages, and you all know that damaged RBCs are also phagocytosed by these macrophages. Okay, then removal of damaged and dying cells and the debris like RBC destruction. Then there is important role is also stimulation of granulopoiesis, because they will release cytokines like interleukin 1, TNF. They will have the stimulatory effect on granulopoiesis. Okay, so tomorrow also we will discuss it. So the macrophage, this is they are the part of the innate immunity. Okay, now what is innate immunity we will discuss tomorrow. So they will help the other cells to develop, means by releasing the cytokines. Now they are interaction with antigen and lymphocytes during immune response, and antigen presenting cell, that we will discuss tomorrow. Okay, so all these are the functions of macrophages and monocytes. Now there is another term called as reticuloendothelial system or monocyte macrophage system. So what is this system? So it is a combination of monocytes, motile macrophages, fixed macrophages and specialized endothelial cells in the bone marrow, spleen and lymph nodes. So all these together they are called as reticuloendothelial system or monocyte macrophage system. Now why it is important? Okay, because whenever there is any infection, from where the organisms will come? Either from the GIT. Okay, so once we eat the food, in that food there will be so many microorganisms, so they will enter through GIT or respiratory tract. Okay, so because we inhale so many organisms, also from the vagina, from urinary tract, okay, skin. So all these are the sources, all are the places from which microorganisms can enter inside our body. Now all these places, especially the liver, because whatever the blood is flowing from GIT, it will go to the liver through the portal vein. Okay, also that from the all the tissue, whatever lymph flow is there, it will go to the lymph nodes, spleen. Okay, that lymphoid tissue. Also whatever is we are inhaling, that will that air will come through the alveoli. So in alveoli also we have the alveolar macrophages. So all these places are lined. In the skin we have histiocytes. So all these places are lined by this tissue, by this monocyte or macrophage. Okay, now they are very protective. So what are the examples of this reticulocyte system? So these are in liver we have Kupffer cells. So these are nothing but macrophages, but they have given separate names. So in liver we have Kupffer cells, because the portal blood, the blood from the portal vein, it is coming from the GIT. Okay, so from GIT the absorption will take place of the nutrients, and the microorganisms can also breach the lining of the GIT. If there is any ulcer or if there is any other problem, then microorganisms can enter inside the blood flow. Okay, then that blood passes through the liver. In the liver, in the sinusoids, we have Kupffer cells. These are the macrophages. They will kill the bacteria, and so they will not allow the entry of this bacteria into the systemic blood. In the lungs we have in the alveolar macrophages. So they will kill the bacteria in the alveolus itself. So they will not enter into the in the systemic means in the system. Now in the connective tissue, beneath the skin we have histiocytes. So if there is any breach in the skin, if there is any ulcer or the trauma, then these histiocytes will play a very important role to prevent the entry of bacteria. Now whatever bacteria which are present in the tissue, they will pass through the lymph node to the lymph to the spleen and lymph nodes. In spleen and lymph node again we have the macrophages. Okay, in the bone marrow we have osteoclast. In the synovium we have type A cell. Okay, like this we have this monocyte and macrophage system everywhere in the body. and at each place they will perform the very important function of phagocytosis. Okay, and they will kill the bacteria and they will not allow the dissemination of these microorganisms in our body. So can you understand, if there is a defective phagocytic system, so they will not be able to phagocytose them effectively, and there are chances of infection, that means spread of systemic infection in these patients. So now in which condition we have monocytosis? So the conditions are tuberculosis, okay, subacute bacterial endocarditis, malaria, leukemia and syphilis. So in all these conditions, because the monocytes play very important, or macrophages play important role in the phagocytosis of all these organisms, subacute bacterial endocarditis, all these diseases. So in these all conditions there will be increase in the monocyte count. So normally it is only 2 to 8%. So in all these conditions it will increase. Now in which condition there is monocytopenia? So again, because of the glucocorticoids and hypoplastic bone marrow. Now let us see about basophils and mast cells. So they resemble with each other, but they are not identical, and where they are present mostly along the beneath the blood vessels. Okay, because the most important function of basophils and mast cells is to release heparin. Now heparin is the substance which will prevent blood coagulation. Okay, now it is very important to maintain the blood in fluid state. So the heparin plays that function. So they are present along these blood vessels in the tissue. Okay, now they are also important in the allergic reactions, because their granules contain heparin, histamine, proteases, serotonin, okay, slow reacting substance of anaphylaxis. So all these mediators, they cause increased blood flow to the tissue, they increase the permeability of the capillary membrane. Okay, so they are involved in allergic reactions, all these mediators. So these basophils and mast cells are that is not the beneficial function, but they are involved in the allergies, hypersensitivities as well as anaphylactic shock. So the conditions in which basophil count is increased is allergy, tuberculosis, chickenpox and leukemia, basophilic leukemia, and on administration of glucocorticoids, and when there is bone marrow depression, so basophil count will be less. Okay, now let us see what is leukemia. So there are two types of leukemia, lymphocytic and myelogenous, based on which cell is affected. So if lymphocytes are affected, then it is lymphocytic, and if other granulocytes are affected, then it is myelogenous. Okay, so in lymphocytic leukemia there is cancerous production of lymphoid cells, and it begins in the lymph node or other lymphoid tissue and then spread to the whole body. In myelogenous leukemia, there is cancerous production of myelogenous cells, and it begins in the bone marrow and then spread it to the whole body. Now what are the effects of this leukemia on the body? So most common is, as it is a cancerous production of cells, okay, it is unlimited, unregulated. So there is invasion of the surrounding bone, which leads to pain and fracture of bones. Also it will displace the normal functioning bone marrow, okay, and it will be occupied by the cancerous cells. So the normal production of cells will be affected. So there will be severe anemia, as platelet count is less, there can be bleeding tendency, as normal functioning WBCs will be less, so increased chances of infection. And also these are the unregulated growth of these cells. The nutrition of other healthy tissue will be affected, and the patient become cachexic. Okay, you must have seen the patients of cancer, because all the nutrition that will be taken up by this cancerous tissue for its growth. So the healthy tissue will be deprived of all the nutritional factors, and there will be weight loss and all these things. Okay, so that is about the leukemia. So that is all about today's lecture. So please go and read the chapter in Guyton. It is given beautifully. Okay, any question?
Ma'am, how does increase in glucocorticoids decreases the cell count?
Because the glucocorticoid they have the inhibitory effect on granulopoiesis. So on these cells, but mostly in eosinophils and basophil, and that's why in allergic conditions also we give steroids. You must have noticed if there is any allergy, then we give steroids because they decrease the number of basophils and mast cells and eosinophils. All are involved in allergy.
Okay, ma'am. Thank you, ma'am.
ठीक है? Okay, now we will meet tomorrow.
Ma'am, do we have class tomorrow?
Yes, we have 4 physiology classes tomorrow. Please come for the class, means please log in for the class because tomorrow's lectures are very important. Okay, you can celebrate afterward after the class, but please log in for the class. ठीक है, चलो बाय.
बाद 14 नंबर दे दो उसको, बोलो लाइन पे ले लो।